Hi everyone, in this session we will discuss about the Silwal inhibitor. Silwal synthesis inhibitor or Silwal inhibitor are those antibiotic which is used to kill or to inhibit the synthesis of microorganisms such as bacteria. The Silwal synthesis inhibitor, there are three different types of Silwal inhibitor. First one is beta-lactam antibiotic, the second one is non-beta-lactam antibiotic, and third one is miscellaneous antibiotic. The beta-lactam antibiotic. Beta-lactam antibiotic are those antibiotic in which the beta-lactam rings are present in their structure. The beta-lactam rings are present such as this is the beta-lactam ring. This beta-lactam ring will be present in the beta-lactam antibiotic. On the other hand, the non-beta-lactam ring antibiotic. Non-beta-lactam antibiotic. Non-beta-lactam antibiotic are those antibiotic in which the beta-lactam ring, this is the beta-lactam ring, are absent in this type of antibiotic. And third one is miscellaneous antibiotic. Miscellaneous antibiotic such as isoniazid and vancomycin. These two antibiotics are present in this miscellaneous antibiotic category. The beta-lactam antibiotic are the most important antibiotic in which the beta-lactam ring are present in their structure. The beta-lactam antibiotics such as penicillin. Penicillin are the most important antibiotic in which the beta-lactam ring are present in their structure. So here first of all we will discuss about the penicillin, that what is the mechanism of action of penicillin against the Silwal of the bacteria. Penicillin. Penicillin are beta-lactam antibiotic, as we know that. The penicillin mechanism of action toward the Silwal of the bacteria. First of all, we will discuss about the structure of the bacterial Silwal. For example, this is the structure of bacterial Silwal. This is the outer layer of the peptidoglycan. This is the peptidoglycan layer of the bacterial Silwal. These are the N-acetyl muramic acid. These are the N-acetyl muramic. NAM represent the N-acetyl muramic acid. Second one is this is the NAG, which represent the N-acetyl glucose amine. The lower side are also NAM and NAG are present. This is NAG, N-acetyl glucose amine will be present here. On the surface of this peptidoglycan layer of the bacterial Silwal, this is another structure are present, which is the penicillin binding protein. This is the penicillin binding protein which provide the receptor site for the penicillin beta-lactam antibiotic. Here some structure are present inside this peptidoglycan layer of the bacterial Silwal. These are some carbohydrate residue arise from this N-acetyl glucosamine and N-acetyl muramic acid. This is another carbohydrate residue which is arise from the lower side of the NAM and NAG. This is the carbohydrate residue. This is also the carbohydrate residue which is present in the lower side of the peptidoglycan layer. Here the peptide bond are present, peptide linkage are present between the carbohydrate residue of upper side N-acetyl uramic acid or N-acetyl glucosamine and between the lower side. This is the carbohydrate, this is the peptide bond or peptide linkage. Here the most important enzyme are present here at the center of the peptidoglycan layer of cell wall of the bacteria, such as these certain type of enzyme are present here and here at the center. These enzyme are autolytic enzyme. These are autolytic enzyme such as autolysine. Autolysine enzyme are present in the center of peptidoglycan layer. So first of all, when we introduce the penicillin toward the patient, so the penicillin will act on this receptor side such as penicillin binding protein. First of all, they will act on the penicillin binding protein side. The penicillin binding protein will activate this enzyme. They will activate the autolytic enzyme, the autolysine enzyme. When they activate the autolysine enzyme, this autolysine enzyme will break the peptide linkage which is present between the N-acetyluramic acid and N-acetyl glucosamine. So this enzyme, this autolysis enzyme will be activated by the penicillin binding protein, and they will break the peptide linkage, the peptide bond which is present between the layer of the cell wall of the bacteria. So they will break the structure of the cell wall. So the cell wall synthesis will be stopped of the bacteria. So here another most important enzyme are present here at the surface of this cell wall layer. Another most important enzyme are present which is called the beta lactamase enzyme. Beta lactamase enzyme are present. So this cell wall of the bacteria, this bacteria will also provide the resistance against this penicillin, this beta lactam antibiotic. So sometime this beta lactamase enzyme will provide resistance against this penicillin. This beta lactamase enzyme will break this penicillin antibiotic. They will break by the beta lactamase enzyme. And other resistance are also provided by this bacteria. Sometimes modification are occur. Modification of this binding protein will occur. Modification will also be occur. With the overdose, with the overuse of the penicillin antibiotic, the modification of this penicillin binding protein will occur, and they will not provide receptor site for the penicillin. So penicillin will not work. And sometimes some gram-negative bacteria will produce will narrow their furines. Some gram-negative bacteria will narrow their furine, so the penicillin will not pass from this narrow furine side. So that's all about the mechanism of action of penicillin. The penicillin are effective against gram-negative bacteria and gram-positive bacteria. In most they are effective against gram-positive bacteria, because in the gram-positive bacteria there are only single layer are present, single peptide layer are present, and in gram-negative bacteria there are two layer are present, outer layer and inner layer. So they are most effective against gram-positive bacteria. The resistance provided by the bacteria with the help of beta lactamase enzyme. So there are certain bacteria such as Haemophilus influenza, Shigella, some Staphylococcus aureus bacteria are produced this enzyme, beta lactamase enzyme, against certain type of penicillin, and they will stop the penicillin mechanism of action. Now we will discuss about the beta lactamase inhibitor inhibitor. There are certain type of beta lactamase inhibitor which provide the best mechanism of action for the penicillin. Some beta lactamase inhibitor are decoy molecules such as leucolonic acid or other are sulbectam, tazobactam, etc.